as many of you know, the WPC's been going almost 20 years now, and was set up really as a way to bring together all the different stakeholders into one forum so we could discuss aspects of Parkinson's. And that's what this Research Spotlight's about. It's to try and take someone who's a very eminent person in that field, who's contributed enormously to find out a little bit about themselves, not just about their work, but themselves, where they began, how they ended up in Parkinson's, and then discuss something about their work, where it's been, where is it now, and where's it going. And so I'm very, very fortunate to have Hilal Lashuel with me today, who is in Qatar, and we'll find out a little bit where Qatar is shortly. Hilal has been, a major player in Parkinson's, done much around many aspects of it, but particularly alpha-synuclein, which is this protein that many of you would have heard about associated with it. So we're going to have a conversation a little bit about that, discuss a little bit about alpha-synuclein, why is everyone so interested in it, and should we be interested in it? But you're very welcome, Hilal. Thank you very much. So just tell
Thank you for having me.
Where you are at the moment and where that is in the world.
Normally I am in Doha, Qatar, but today I'm actually in Geneva, Switzerland.
All right. But you're based in Qatar?
Yes, I recently moved from the EPFL in Lausanne as now a professor of neurology at Weill Cornell Medicine, Qatar. I'm also the research development and innovation advisor for the chairperson of Qatar Foundation.
Okay. And so that's a wealthy foundation that supports medical research, or is it just Parkinson's?
It's a foundation that was founded 30 years ago in Qatar to lead as an engine of transformation for the country through research, education, and social development. One of our biggest projects is Education City, which is a 12 square kilometer campus that has 8 universities, 3 research institutes, 4 policy centers, a women and children hospital, and most importantly, 13 K-12 schools. So it's an entire ecosystem in one campus.
Okay, so that sounds terrific. Well, perhaps we'll come back to that at the end to see how that has invigorated and taken your research on to new levels. So, I mean, where did you begin life? Did you begin life in Qatar and gone back there, or where did you start, and how did your career? What did you do early on in your career?
So I'm originally from Yemen. I left Yemen at the age of 15 years old to New York City with my 16-year-old brother alone. We lived in Brooklyn, and
which is where Eli lives, I think.
Which is where Eli lives, and I came from a background. We were the first from the family to actually go to school, let alone go to college.
Right.
We landed in New York alone, and basically at high school we had to work about 30 hours a week as young people to put ourselves through school and pay for our life and education. I started my undergraduate in the field of chemistry. I originally went to the US to study medicine. So in the second year of my pre-med education, I started to do research as a volunteer in an organic
So was this in New York as well?
This was at Brooklyn College, City University of New York.
Okay. Yeah.
And it was at that time, we didn't have growing up role models or people to tell us a lot about college and how to choose and what to do. So everything, I discovered on my own, stumbling, standing, and back and kept going. But it was as an undergraduate that I basically discovered my passion for research. I knew at that time I wanted to be a researcher. I didn't know in what field. I had specialized as a pre-med in chemistry, so I continued in chemistry. I got my degree with research in the field of inorganic chemistry. And then I started my PhD degree at Texas A&M University, working as an organic chemist and a researcher in a lab. And the focus of my research, so I got into the neurodegeneration field by accident, actually, because I started my research trying to develop model systems using chemistry to study actually how protein folds. What are the kinetics and thermodynamics? What are the factors that control protein folding? And I was focusing on one very specific type of folding, which is proteins that are rich in a specific structure called beta sheet. And the challenge I had is every
So
time
these beta sheet proteins, these are things like amyloid, which you get in Alzheimer's disease.
it's
These are
like
quite common in
amyloid,
the
yes.
human brain
So
with
yeah.
disease.
So
Yeah.
most proteins contain a mixtures of different structures, and beta sheet is one of them. But proteins, these beta sheet structures are quite contagious, so they can line up, stack. But what proteins do is they do a good job in hiding that part by other structures so that you don't have this linear assembly.
Okay.
And in my model systems, whatever I kept developing kept self-assembling and making what we call these filament-like structures that look like the amyloids you have in the disease. Now, half of our lab was working on studying an amyloid disease that's called systemic amyloidosis and familiar amyloid polyneuropathy, which is caused by the aggregation of proteins that deposit in the heart. So after 3 years of not being successful in my PhD project, I figured I have to make good use of what I learned, or what we call when life gives you lemon, you make lemonade. So I took the experience I learned in terms of how to study these self-assembling model systems and started to apply them to study, the misfolding, the assembly of proteins that are associated with disease. Focusing.
So can we just,
Sure.
at this point now, obviously this is, extraordinary story that you arrive in New York, basically you and your brother, it's a sort of, you arrive, you've got to find yourself somewhere to live, you've got to build your life there. And did your brother follow a similar line? Has he gone into a similar sort of area, or did he move into something?
He's not in science. My father went to education, first geology, and then he specialized. He has a PhD in education today, and he's an executive director of a big institute.
So you're the first person in the family to sort of go to university, get a PhD, and then do research, really.
That's correct. First researcher in the family.
So it's interesting because a lot of people we've had on this, Research Spotlight, they've had parents or family members who've been in research or academia, and they've sort of followed. But for you, this was like a huge new adventure which nobody in the family had done. And the reason you moved to Texas was because the opportunity presented itself, or they had the project in organic chemistry?
They had a very well-known chemistry department, so I was interested in chemistry. So when I started my PhD, I wanted to redirect my work toward more biology rather than inorganic chemistry. So I joined this group, and then I focused on switching more to disease-related project. But our focus was still trying to study the disease from a perspective of a single protein. Trying to understand. At that time, we knew that most of these diseases, including Alzheimer's and Parkinson's, are characterized by the presence of these aggregates. But at that time, we didn't know what the mechanism is. How do we go from a normal functioning protein to this disease state?
Yeah.
And I spent
what led you to that transition? So obviously, the lovely thing about chemistry is it's quite nice and clean. You have one molecule, another molecule, you stick them together. And, here in Cambridge in the UK, we have a lot of chemists who think they've cracked Alzheimer's disease and Parkinson's disease many times because they just make proteins, put them in test tubes. But why did you move away from, if you like, pure chemistry to slightly more applied chemistry? Because obviously you got these filaments when you were trying to make proteins in the lab. And then you decided you want to actually move into the biology of the chemistry of these proteins. So what triggered that? Was there somebody you met with a condition, or was it just that you felt I wanted to be more biologically
initially, as a PhD student, I was more interested in a biological problem that has a disease relevance.
Okay.
And then, we still approached it from trying to understand this mechanism and understanding which form of these aggregates is the toxic species. Once I finished my PhD, I started a position in New York. And then I began to feel the limitations of our reductionist approach, which is reducing a protein, in the sense that as a protein biochemist, I was able to come up with a lot of mechanistic models and hypotheses, but I didn't have a path to test them. And some of the ideas we had were crazy, and some of them did not make sense to biologists. So it was always very difficult to convince people to do the experiment for you. So as I started to build my research team at the EPFL, I had that goal from the outset, that I need to start in areas where we're strong, that is biochemistry and biophysics. But it was always the target to try to build in that pipeline that allows us then to go and assess and test the biology. So we
So
started
from
to work a
Texas,
lot.
did you go back to New York then and start working with biologists there, or did you
So
sort of carry
after
on?
Texas, I had a position at the Picauer Institute to work on Alzheimer's disease. That's when I started working on
And where was
Alzheimer's.
that?
That's in Long Island, New York.
Long
But I
Island.
was the only one leading the program. There was no critical mass around
Okay.
me, and I felt didn't have the biological context. So that's when I went to work with Peter Lansbury at Harvard, and for a postdoc. And my goal there was really to learn the biology of the disease.
Yeah.
But when I got there, there was an interesting structural problem, and I remained focused mainly on biophysics and structural biology. But my time at Harvard was transformative in two aspects. One, even though that we continued to do biochemistry and biophysics, we were always surrounded by clinicians. That's when I started to look at the disease from the clinical perspective and to look at the disease from the neuropathology perspective. From there on, everything what we do, even applying reductionist approaches, was really driven by the biology of the disease and not by an interesting chemistry question. And that's how
And,
we oriented our work.
and so were you, I mean, I guess, so this would be in the sort of, what, 1990s,
This
early
would
part
be
of
early 2000s.
early 2000s. I mean, what, what role did chemists play in the world at that point around neurodegenerative
Oh,
diseases?
I think
Was it regarded as a bit of a fringe subject, or was this an area? Because I could see the problem. You're working in chemistry, biophysics, how these things sort of behave, and then you've got biologists who work with cells and animals and human specimens, and there's a sort of, how do we talk to each other and how do we
yeah.
work?
So
So were you
it's
regarded as a bit of a
interesting.
freak in those days?
Yes, I mean, I must tell you, because we were the resolving mechanism at the level of a single protein,
Yeah,
and then I would go to conferences where people talk about animal models,
yeah,
and it was always very awkward because people looked at this and say, this is just a test tube effect,
yeah,
and then eventually I learned, that mouse, rats, and flies are also not human, so I was able to defend myself. But the most important thing is, I think if you look at the field of protein aggregation, that the
yeah.
most fundamental biophysics were actually defined by people in biochemistry and biophysics,
Okay.
in terms of the mechanisms of aggregation, the kinetics, the structural aspect. Much of the tools we have today to study this process were developed in those labs, not in the biology labs. And for a while, there was this disconnect until
Yeah,
we got to a point where it was not possible to explain things by what you see in animals. For
yeah,
example, the fundamental problem was the lack of correlation between the pathology you see in the brain and the disease. And
yeah,
then people couldn't explain it. And the only way was to explain it was to say, to go back to fundamental biophysics mechanism, which is there is something between the native protein and the aggregate, which are the oligomers. And then the
okay.
oligomer hypothesis became very popular because it explains things we couldn't explain in biology. So
So
that's
basically, just to get this pathway, because I'm not sure everyone's entirely familiar with this, and I'm one of them. I mean, so essentially, the sort of problem, I guess, in diseases, and let's take Parkinson's, given this is the WPC, is we have a protein, alpha-synuclein, which is 1% of our brain. We all have alpha-synuclein, we have tons of it, and it's clearly doing something that's very important. And then we have people with Parkinson's, where the alpha-synuclein has aggregated, and you can see it in the brain when they've died with these clumps called Lewy bodies of Lewy neurons. So we have this aggregated form and we have the normal form. And I guess what we're saying here is that for some reason in Parkinson's, for most people who don't understand why, this normal form slightly changes. And it changes in a way that we're not quite sure. And I'll come back to this in a minute so you can help me understand it. And I guess the assumption was we had normal alpha-synuclein, then we had aggregated alpha-synuclein, and what was in that aggregate was clearly what was the problem. And what you're saying is actually that there are intermediate stages between the normal and the abnormal. And because we couldn't see the abnormal, we were sort of missing it and saying we're actually not looking at the right protein here. We may be missing a species, which is actually important. And I guess that's where the chemistry comes in, because you can obviously you could presumably make a molecule of alpha-synuclein and then you can modify it and you can see how it behaves. So is that the sort of basic theory correct? And then the other question I want to come to you about is, one of the things which I always find interesting in chemistry is that people look at the biophysical properties of protein. So you put a protein in a test tube, you add a bit, you change the temperature, you shake it, a bit of cookery, and then it all aggregates. And then you plot it all and it looks terrific. But that's hours, possibly a few days.
Yeah.
And you think, I'm looking at a condition which progresses over years and decades. So is that why the biology is so important? Because what happens in the test tube gives you insight, but the kinetics, the speed with which things happen are so fast that it couldn't really apply to the disease because it's too quick.
I think the advantage of the reductionist approach is you always work under controlled conditions. And as a chemist, everything is always a reaction. There is a reaction and products. Yeah. And you working under defined conditions with very defined material, you can ask very specific questions. The reality is that the biology is complex.
Yeah.
But in a sense, you have to start somewhere. What is important is not to get trapped in that and to avoid the biology because this is complex. And this is sort of, I think, a price we paid for years. We didn't pay attention to the complexity of the biology, and we were limited by technology. To give you an example, we now know that for at least for Parkinson's disease today, that the structures of the fibrils or the filaments found in we produce in the tube do not resemble those that exist in the brain. They're structurally different. And what we produce in the test tube doesn't have the same chemical decorations, the same modification, the same alteration as what you see in the brain. So the standard argument would say, I can use this reductionist approach to study the mechanism, but the way I do this should be informed by what I know from the biology of the disease. And I shouldn't just study this because it's easy to study, because from an academic point of view on mechanism of aggregation, it's okay. But when I start to talk about developing drugs and diagnostics, then I need to find a way to approach the real biology and pathophysiology of the disease. And that's what we tried to do in our work, especially over the last 10 years.
So I guess the strength of your approach is this reductionist approach. So if I make a form of alpha-synuclein, whatever it be, I know how it will behave under certain conditions. The question is that what's happening in Parkinson's? But it gives you an insight.
There's no green thing on these things.
I think someone's.
I think the way.
So basically, you can say that's how it behaves. The question is, how faithful does that resemble what I see in the brain? So, well, it doesn't. It gives me insight. I can see that it aggregates. Exactly. And so now, what's different about the patient, which I can then take back to the lab? So it's this dialogue.
To give you a simplified example, we know that the majority of synuclein in the brain, in terms of the aggregated form, is phosphorylated.
Yeah.
But we studied everything using unphosphorylated protein. And the problem, and there are other modifications like ubiquitin and others. Yeah. So for a long time, our lab, we were working with a protein that is what we call naked, doesn't have anything in it. And then we realized it's not the same. So when we started to tell people we would like to reproduce what's in the brain, there were 43 forms of synuclein that have been reported. And the question is, where do you start? And if you ask the cell to make it for you, the cell will give you something else. So what we did in the lab is we spent 4 years developing a method where we build synuclein one amino acid at a time. It's called, so we synthesize synuclein completely in the lab. And that gave us the freedom. So in a matter of 12 years, we actually reproduce the entire proteoform. We can make every single form of synuclein that exists in the brain. So that was our approach of trying to, what I call, embracing the disease complexity and bridging the gap between, in vitro systems and the reality in the brain.
So just to sort of explain that, in case people don't understand the sort of details. So basically, if you make a protein in the lab, that protein will just stay in that form because you just made it. It's just like if I make a shelf and I just leave it here, it's just going to be a shelf. Of course, if I put it into something, i.e. the alpha-synuclein into a cell, the cell will modify that protein. So it will add these phosphate groups, so phosphorylated, or it will nip bits off, or it will add a bit of some other modification somewhere else. So the cell has the capacity to modify the protein, which you'll never see if you just look at the protein on its own. But what you did is you said, well, let's look at the brain. There are 43 different types of alpha-synuclein. I've no idea whether any of those are relevant for Parkinson's. And then every protein is made up of amino acids. So you knew the structure of alpha-synuclein, you made it, and then you put each of these modifications, and then I suppose you now have a library of 43 forms of alpha-synuclein. You can say, well, which one would you like me to study, under which conditions, and then how does it behave? And I guess that then gives you clues as to which. So if we're talking about aggregation, I guess some of them aggregate more quickly than others. Some of them doesn't seem to make much difference.
So we were surprised. We were totally surprised, because we found that most of the modification that characterize the disease states do not promote the disease state.
Yeah.
So because we have each modification at the level of when we ask, do they make the protein aggregate more or less? And we found that the great majority either have no effect or this, which means that, which we can show later now, that these modifications do not drive the disease, but control the late stages of how these pathology are formed, how they're cleared, and how they interact with other parts of the cell. And we would have not been able to do that if we didn't go to the chemistry approach, and we didn't go back from the reductionist, but then back into the biology. And the second thing we tried to do is once we worked at the protein level, then it has become clear over the years that the pathology, the Lewy bodies, the Lewy pathology, it's not just proteins. It's a whole gamut of a structure that has proteins, dysfunctional organelles. And the question is, how do they come together? So then we went from chemistry to use cell biology to reverse engineer the process of pathology formation. And it's quite remarkable. You talked about the time difference, that in a neuron, and now we could do this in human-derived neurons, we can actually reproduce similar pathology to what you see in the brain in terms of protein composition, in terms of organelle composition, in terms of the type of aggregates. So despite the short time, but Parkinson is more than pathology. It's more than neurons. It's how neurons interact with other cells. It's the circuit level interaction, and it's a degeneration process.
So let's pick up on that. So I wanted to start off by saying, how do you know it's 43? How do you know it's not 83 and you've only found 43 of them? So how do you know?
Well, the way we did it, this is a very good point, because today we tend to study Parkinson's only based on the tools we have.
Yeah.
So, for example, if you look recently, the way we study pathology is mostly with one antibody, which is against phosphorylated synuclein. And that's because it's not the only form, is because we didn't have the other tools. We didn't study oligomers because we actually have no tools that can specifically detect oligomers and distinguish them from the other forms. So we always look at the disease through specific lenses that are available. And one of the strength of the approach we applied in the lab is what we did is we said, let's look at all forms of synuclein that have been reported to exist in the brain and biological fluid. And then what we did is we reconstructed them at the level of proteins, but then we made a library of about 48 antibodies that can capture that diversity, and then use that antibody to go back to human brain. And what we showed is that it's very difficult to capture the full human pathology with a single tool. And that we need to move to these expanded tools, because the pathology comes in very different flavors and different shapes, which are not detectable by a single antibody and tool.
Okay, so if we go back, so you've defined. So we say alpha-synuclein lies at the heart of Parkinson's disease, and the story of that goes back to the families, then finding it in Lewy bodies, and then other genetic triplications of the alpha-synuclein gene causing Parkinson's disease. So alpha-synuclein clearly lies at the heart of it. You've defined the types of alpha-synuclein that exist in the brain. You've then developed an array of antibodies so you can stain it in the human brain. So if we go back to those days, we now know all these different types of alpha-synuclein. We now have some suspicion that some look as though they could be causing more problems than others. So the next question I have for you, Hilal, is how do you then study that in a system? You say you look at it in cells. And the question I have is how dynamic is this process? So I think one of the problems we have is we often think a bit of alpha-synuclein does this, then it does that. And probably in all of our cells all the time, this is a very dynamic process. So how do you study it in a biological system? What system would you use
So
And
the
then
way we
how
yeah,
dynamic is it? Because
yeah.
obviously for most of the time, it's not causing problems.
So the way we and most people in the early years used to study the role of aggregation is to make different types of aggregates and add them to cell and see what they do to aggregates.
Okay. And these aggregates are artificially made in the lab, or you take them from Parkinson's brain?
Yeah. And then when it became clear to us that there is differences between what we produce in the lab and what's produced in a living cell, we had to work on sort of developing model that reproduce the entire process. And what we found through our work is that the number one thing is it is not necessarily the aggregate itself, but the process of aggregation.
Yeah.
And the first experiment, we showed that, for example, if we, let's say, take fibrils and add them to neurons.
So so fibrils they're not oligomers and they're not aggregates. They're somewhere in between, aren't they?
Fibrils are the most mature form, and they have robe-like, filament-like structure. They are an end stage, whereas oligomers are small aggregates that are still soluble and can float in solution, are not precipitated. And then
aggregates are just made up of fibrils, are they? They're just
aggregates, Lewy bodies are highly rich in fibrils. They're all the core structure.
Plus all these other bits you were telling us about.
So what we found is first, when we add these fibrils to cell, they sit in membrane and they don't do anything. If we add this more monomeric subunit, then the fibril starts to grow. And as it grows, it actually sequesters lipids from the membrane, causing the disruption of the membrane. So this is an example of how the process, rather than the aggregate itself. So the dynamic of the process is so important.
So you have your membrane here, the fibril binds to it. You then add basically building blocks to it. It then grows. As it grows, it pulls the membrane out. The membrane dysfunctions. And because membranes make up mitochondria and other things in the cell, it all starts to fall.
So this is the first time we began to think that we shouldn't think about just a specific aggregate entity, but the process of aggregation. And the classical example of this is, we very often have to deal with the question of why there isn't a good correlation between fibril burden in the brain and the disease. And the argument we use is because the damage is done on the process of forming the fibrils. So if we take fibrils and add them to neurons, they get in about 80% of the neurons, but they do nothing. The neurons are happy if they don't express synuclein. If you have a neuron that expresses synuclein, then these fibrils grow. And this growth process is also linked to the interaction with organelles, causing other organelles to dysfunction. So that's where I say we have to look at the whole process and not. And if we start to think about this in terms of process, it helps us deal with the discrepancies that we see in pathology. For example, the question is always, do the Lewy bodies in the brain, are they protective or are they toxic? And my answer is they don't have to be either. They could be both. Because if you think about it from a process point of view, what's in these Lewy bodies? They're aggregated toxic proteins. They're dysfunctional organelles. So if the process is efficient, it's like groping, then it's protective. You're taking everything that is toxic and containing it.
Putting it all in a trash can.
But if the process is defective, because the process of packing and reordering, then it can become a toxic process because everything now in the cell. So what we say in the field is it's the dynamic of the process that is important rather than the specific entities in that process.
And so the hypothesis would be it's the process of the growth of the alpha-synuclein from these fibrils that's damaging the cells. And the reason the cells die, just to be clear, someone's asked a question about mitochondria, but within every cell we have these organelles, mitochondria, lysosomes, ER. They all are made up of membranes, and so as the alpha-synuclein is sequestered or growing inside the cells, it pulls membrane from wherever it is. That makes all of these organelles unwell, and together they cause the cell to dysfunction and then eventually die. And in the process of that, parts of that membrane get caught in the fibrils, and that's what forms the aggregate. And that's what goes wrong in Parkinson's. So you would say the fundamental problem in Parkinson's, the first event is the growing or the degeneration of some oligomers and then fibrils, which set off a cascade of events, which then leads to the cell
I think,
dysfunction.
It's never as simple as that, but what we've learned from our work and what we learned from our understanding of human pathology, it's not just the fibrils, it's how the process of fibrils and the fibrils interact with all the cellular components that matters. So that's something we haven't factored in before, because we were always studying fibrils alone and developing therapeutic based on looking at them in, detached from this reality. Because, for example, we have data to show that this process of growth occurs on the surface of the membrane structure of organelles like mitochondria and lysosomes, causing their the dysfunction of these organelles, which then contributes to neurodegeneration.
And do you think this happens normally? But normally we're quite good at reversing it or stopping it, so the cells survive. So one of the questions is, how much is this a normal process, or do you think this is unique to Parkinson's? And then the second question is, if this is the process, why don't all the cells do it? Why doesn't the whole brain go down with this?
So the question is, because aggregation is a process, you need two things. You need a trigger. And you need an environment that is conducive to self-assembly. So the first thing that happens is you have a protein doing its normal functions. For example, synuclein spends a lot of time on the membrane. Then there's something that has to happen. It's either due to a genetic mutations, to a stress conditions in the cell, exposure to chemicals or pesticides. That protein is began to change its form. And now, normally, the cell has a very efficient quality control machinery to recognize if a protein is no longer in an abnormal state to clear it. But somehow in aging, or if this process is overwhelming, the cell has difficulty to deal with it. And then once you make the first types of aggregates, the smaller fibrils, the process becomes autocatalytic. So then thermodynamics is against you, because you only need a small amount to keep the process going. And
so once it starts, it sort of self-propagates.
So some cellular environment are more conducive for this process than others, and that's why we have this specific cellular vulnerability in different diseases. Because Huntington may not aggregate the same place as different proteins prefer to aggregate in a specific environment.
And this, whilst we're talking about alpha-synuclein in Parkinson's, you've mentioned Huntington's, which is a different disorder, genetic disorder, but again, there's a protein that aggregates. And it's the same principle. In Alzheimer's, you have amyloid and tau, and then in other diseases, you have tau. Even in Parkinson's, you might have to. So all of these so-called proteinopathies, these diseases of the brain with these aggregates, the principles are the same, different species of protein, but fundamentally it's the same. And so that raises the question, do you think one can trigger the other? So, could there be, so if a bit of alpha-synuclein starts to go wrong, could that then cause tau, another protein, to aggregate? Or is it protein specific?
This brings us to a different, sort of another layer of complexity that is important, because we've always looked at in the past these diseases as a single proteinopathy disease. The idea that Parkinson's only deals with aggregation of synuclein, whereas Alzheimer's, amyloid and tau, whereas Huntington, it's now becoming clear that actually this is the exception rather than the norm, in the sense that most patients, or most Parkinson patients, show aggregates of very different types of proteins, and there are data showing that if you have more than synuclein and other aggregates, they do affect the clinical manifestation of the disease. And which led some to say, can you cure Parkinson dealing by targeting only one protein?
I'm going to come on to that.
So, and the question is, it depends, right? If Parkinson's occur first and then triggers other pathology, then or make other cells more susceptible to form pathology, then you can think that's the case. But today we actually understand very little about how these co-pathologies come up. Do they come up because the whole cellular system becomes weak in terms of proteostasis, maintaining equilibrium of all the proteins because of the stress caused by synuclein? Or is it through what we call direct interaction or cross-seeding? that is an aggregate of synuclein, can actually, induce a seeding effect and induce the other protein to aggregate. And this is still an area that we understand very little about because we don't have preclinical models for co-pathologies that are very good. We're still struggling of making great progress in reproducing each of these individual pathologies.
Okay, so I'm going to come on to treatments in a minute because obviously you have a very clear hypothesis, which in some ways, the ultimate proof of it, can you interfere with it in people and slow down the disease? But I mean, Hilal, when you're trying to model this in the dish in the lab, you obviously talk about the fact you've got these cells, you can see this process. What do you use to trigger it? So, we've had a few questions here about, what might trigger it. Is it immune response? Is it toxins? I mean, there's a lot of interest at the moment in pollutants, these nanoparticles of plastic and whatever. So how do you model the aggregation in the dish in your model systems in the lab? Do you just add fibrils and watch it work through that? Or do you use other triggers which might be associated with environmental factors linked to Parkinson's?
Yeah, so one of the challenges we faced in the field of Parkinson's disease is how do you initiate aggregation of a physiological protein into a disease state? And it became clear that mutations were not enough, right? If you simply make a mutation without changing the level of proteins, it doesn't work. And then we went into the idea, well, in aggregation is a concentration dependent. The more you have of the molecule, the more likely they will aggregate. So we moved into what is known as overexpression models. You just bring mice that express high level of these proteins with the idea that they will go. And in all these models, we made some progress in the sense that we could produce an aggregate. We could produce aggregates that have some feature, but we couldn't reproduce bona fide disease aggregates. And actually, today, there isn't a Parkinson model that goes from a normal physiological state to a Lewy body without external, simply by exposing to a toxin or modulating the immune response. The best models we have now, so the first step seems to be the most critical step. How do you form the first aggregate? So the breakthrough came with the concept of seeding. That is, you overcome that thermodynamic or entropic barrier by adding a preformed seed that you get from a human brain or from a test tube into the cell. And what we found is that this is work started in Virginia Lee's lab and others. If you take a preformed aggregate, you add it to the cell, it goes in fast, and then it actually triggers aggregation. And then we get the formation of filaments, which actually for years people stopped at that stage. And what we showed is that if we wait longer, we actually get to a Lewy body-like structure. But the disadvantage of these, what I call preformed fibrils or seeding models, is you're able to get to the pathology, but now you're missing the first phase, which is how do you go from monomer to oligomers to fibrils? Because in these models, we start already at the fibrillar state. So it's a good model to look at the role of the process of fibrillization and the formation and maturation of Lewy bodies. But it's not a good model to look at early oligomerization state. But it's the best model for that right now.
So do you think that's a good model for studying? So obviously a lot of people study models by which to look at alpha-synuclein behavior and then say that informs what I'm going to do in the clinic. So are these models useful, or are they, because you could say, well, if I stop the aggregation and stop the Lewy body-like structure forming, then whatever it's doing, wherever in that process it's working, I don't really care because it seems to have worked. But if I've understood you correctly, what you're worried about is a lot of things may not work, but actually that's because you've missed the critical step. You haven't understood the first.
So I think it's very difficult today to have a single model that will allow you to answer all the questions. Because in this model, we form fibrils and we form Lewy body-like structures. It's very useful to study that window of that mechanism. For example, at least now we can form Lewy body-like inclusions, so we can study how do the cells clear them? How does the interaction with microglia and astrocytes in the clearance of these aggregates? What we were able to show through the system is now we can ask which step along this pathway is what causes degeneration.
Yeah.
Right. But what we cannot rule out with this model is what's the role of early oligomerization state. So for that, you need a separate model that allows you to model the early state. The ideal model would be is the one that allows you to go from monomers all the way to oligomers, all the way to fibrils and Lewy bodies. Such models don't exist. So I think in Parkinson's disease, we can always start with one model, but we must do the validation in multiple models before we can go to humans.
And so do you think the models we currently use, I've heard what you've been saying about some of the limitations, but the fibrils which we're adding to trigger this, I mean, a lot of people say that those fibrils aren't actually what you'd find in the disease itself. They're rather artificial. So it shows protein aggregation. It shows a principle, but actually its relevance to the disease must be questioned because it's not what's actually seems to be lying behind Parkinson. I don't know if that you feel that's a true statement, in which case
I think we have to be always careful. The debates in the field tend to always try to decide between two things.
Yeah.
And the reality is that it's not that simple. So I cannot use synthetic fibrils to develop drugs that I will use in a human.
Yeah.
But what we have shown is that the synthetic fibrils, if you add them to cells, they will produce filaments that have the same signature as in the human. So they are sufficient in a cell to trigger the process. And we get to a Lewy body-like structure. So for modeling, they are perfect, But if I would use them, to try to develop therapeutics, then there is a challenge because we know now that the fibrils from the brain are not the same. So if they're not the same, then the biggest question in the field we have today is once I use the synthetic to induce pathology, aggregation in a cell or in a mouse, am I producing pathology similar to the human? Or am I producing pathology similar to the synthetic material? If I'm producing pathology similar to what's in the human brain, then the model becomes perfectly, a great platform for discovery. But if the fibrils we're producing in these preclinical models are very distinct in terms of their structure and biochemical properties, then we have to step back and rethink, how to use these models. And I'm not saying we need to drop them. We just need to know, up to what level the models can take us before we can make disease-relevant discoveries and therapeutics.
Okay. So there are just two things I want to... We're sort of getting into the last 15 minutes. So if you have questions, various people have put them in, so that's great. But essentially this principle you're talking about, amplification templating, this is very much what this, great excitement's been recently about this seed amplification assay as a way of diagnosing Parkinson's. So, there's been quite a lot of discussion about that. This is the idea you can take fluid, blood, CSF, of a lumbar puncture, and you can, from someone with Parkinson's, you can test it. And this amplification, which you've seen where the alpha-synuclein grows, you can do this now and show that someone's got this, in which case they're very likely to have Parkinson's. Do you see that as a major advance in the field and actually highly relevant to, how we see the condition developing? Because it seems to me that assay is very much based on the principles which you've just discussed as a way in which the disease comes about in the first place. So I suppose what I'm saying is if that's true and we treat someone and their seed assay goes negative, then we've solved the problem.
I'm not sure it's that simple. So let me go back and break it down. The seed amplification assay, I think, represents a major advancement in the field because what we're using is the same thing as we're doing for PCR. We've developed a way where it would allow us to detect something that is not detectable. And what this assay shows now, the power of this assay is if you take 3 different types of these assays and conduct it in 3 different research groups on the same set of samples, you have concordance and you have diagnosis up to 90%. The limitation of these assays is right now is they're good at diagnosing advanced stage of Parkinson. They're not good at predicting who will get Parkinson's yet. And the question we have is whether we will always have a single measure as the right diagnostics.
Yeah.
You know, whether it's amplification assay, whether total synuclein. And I think it's going to be a combination of different measures that could probably come up to a better diagnostic. But today, this is a major advance. We have to improve this assay to make it quantitative so that it could be useful for monitoring disease progression, to see response to therapies. My main problem,
because at the moment it's a bit positive or negative. It's not.
I think there is some progress in work by Byron Cowie and others to try to make it quantitative. It could be challenging, but I think there is good promise. My concern as someone who has been in the field for a long time is the assay is quite misused, and abused. The second, there are a lot of labs that don't, it's a very difficult assay to establish, even for a lab like ours with 25 years of experience. It took us a long time. And the major trend of concern is that this assay is now done mainly by companies, and the companies are not making the raw data available. And therefore, you're able, and this is, I think, puts a lot of risk, because we need to ensure rigor and reproducibility. And the only way, and also this data can be useful for optimizing the assay. So we wrote a piece in Science Advances early this year calling for more sharing of raw data that would enable the use of machine learning and AI to improve the quantification of this assay. But I think it's definitely one of the major advances we've seen in diagnostics. And I have a confidence, given what we're seeing now in Parkinson, Alzheimer's disease, that this is an area where we'll see huge progress in the next 5 to 10 years.
Yeah. So it's been a major advance, and it obviously in some ways stems from the work that you've done and others about how we perceive the sort of pathophysiology, the pathology of Parkinson's around this amplification of alpha-synuclein and its misbehaving. So if I come back to the role of alpha-synuclein, there have obviously been a lot of interest in targeting it, and there are various ways in which you can do that. But the most obvious way would be to regard alpha-synuclein, if you like, as a sort of microbe or a bug, and you generate an antibody that kills it off. So you either give someone an antibody to alpha-synuclein, or you get them to induce an antibody to alpha-synuclein, and you kill off, therefore, the species of alpha-synuclein is causing the disease, and therefore you should be able to slow it down and stop it. So, that's the principle of it, and that's been around for about 15 years, and yet none of these alpha-synuclein immunization trials have given us a positive outcome. So is that because alpha-synuclein is interesting but not relevant for Parkinson's? Is it because we've not done the antibody therapies properly? Or we're targeting the wrong species of alpha-synuclein, or what would be your interpretation? Because I often get asked as a clinician, people tell me alpha-synuclein is important. They've done all these trials, they don't work. So why do you still think it's important?
So I think there are a number of factors. The first factor we can't rule out now is everything we've been using to develop these antibodies and these aggregate targeting have not been done using structures that resemble what we see in the brain. We now know that no one is able today to produce in a lab something that has the same structure. Now, these fibrils are also decorated with these PTA modifications, and we don't know how these modifications influence the binding to the antibodies. It's very rarely assessed. Finally, is the concept that.
So just to summarize that, you think we may be generating antibodies against the wrong species of alpha-synuclein, and we don't really know what the species is we should be targeting, because I think the ones we're doing in the lab are not the ones we find in the Parkinson's.
Yeah, for example, many of the antibodies that were initially developed against synuclein were against the region that gets cleaved off in the pathology. So I think the design in the past, we have not designed therapeutic targets that are designed to capture the diversity of pathology. This is one potential contributing factor. Two is, I think, the fact that we start to apply these methods, when the pathology is already spreading in the brain.
Yeah.
And therefore, you're fighting an uphill battle. And if you're not capturing that diversity, you only need a small amount of seeds that continue to propagate on this. The third is, to your point, is whether all the damage is done by alpha-synuclein and whether it's the aggregate. And this, I go back to the process. one has to keep an open mind that the process of aggregation may trigger other processes that drive neurodegeneration, independent of what happens to the aggregate later.
Okay.
And therefore, if you think about that, then the correlation at some point get lost between burden and this. The fourth thing is we published recently several papers showing that it's not merely the presence of the aggregates, it's the properties of the aggregate. So if we take synuclein aggregates, we add them to mice, they propagate and they spread. If we take the same aggregates and we introduce a chemical modification that is like disease, nitration, or adding a sugar molecule, these aggregates become inactive. They don't spread, they don't seed. So what we're doing is we're quantifying everything as if it's one, the bad aggregate and the good. And therefore, I may have reduced aggregates that are actually don't do anything. So I think the simplification that there is one form of aggregates, they all function the same, is too oversimplification, because we know we have brain region where we have pathology that correlates with degeneration and areas where it does not. And people call it both synuclein aggregates, but they could have very different structure and properties. And
so you're saying so we're not quite sure what we're targeting. When we target things and we see changes, they may be in forms of alpha-synuclein which are irrelevant to what is causing the disease, and it may be that different types of alpha-synuclein do different things in different cells, and calling it all the same is actually rather unhelpful. What about the argument that they don't get in? So there's always this question about, are we actually do these therapies really get to where they need to get to in the brain? I don't know.
I mean, this is always the case with any therapy, whether they get to their target, whether they get to the target at the same concentration. And there is always a risk, but I think the limitation on this is having the right assays to look at target engagement. But we actually have a better way of targeting.
Well, I was going to ask you now. So in the last 5 minutes, I want you to tell me how.
Yeah, that's what I want to focus on, because this is what we will be working on in the coming years. And that is, it's an area where actually work I did as a PhD student that ultimately paved the way, along with work I've done to the first FDA-approved anti-aggregation strategy for systemic amyloidosis, for TTR. Okay. And the idea is that, once we go down the line of aggregation, life gets complicated, because what type of aggregates, what modification. So the best way, we can prevent the entire process is what we call by making the normal protein happy. It's a concept we call native state stabilization. That is, we know that the normal state is not, it doesn't cause disease. So if we can develop drugs that keep synuclein in the native form, then it never has to misfold. Then I don't have to worry about which species to target. Now, that approach itself will not be sufficient, because this approach would work if I know that somebody is at risk of Parkinson's disease. But once somebody has the disease, it means they have the normal form and the disease form. So in my view, we will need a combination therapeutic approach, one that focuses on stabilizing the normal form, and one that focuses on getting rid of the abnormal aggregated form so we can reduce the seeding and the spreading to the brain. It's almost as if you have a flood in the house. You can wipe the water as much as you want, but the best way is to turn off the faucet. So you need to be able to turn it off, and that starts cleaning the mess. So I don't think, and this is something we need to engineer into our approach, that the monotherapy would actually work for the different stages of the disease.
But in systemic amyloidosis, it works. I mean, they take quite a reductionist approach there, but that's because typically people have a mutation or an abnormal gene, and you switch off the production of the abnormal amyloid. Is that right?
No, actually.
For these, or is this for? This isn't for familial amyloidosis.
No, so the way it works in systemic and familial amyloid is that protein is tetrameric. It's a very stable, has 4 subunits, and it has a binding site for thyroxine, T4. And what we showed is the work, it says my PhD, is that for this protein to aggregate, it first has to dissociate to a monomer, and then it changes shape and aggregates. So if we can keep the protein in its native tetrameric state, it would never aggregate. So the drugs we took is we started with a native substrate, which is T4, and we showed that if you put thyroxine there, the protein never aggregates. And the other hint was TTR, the protein, is the main thyroid hormone carrier in the brain, but not in the body, and there was no amyloid in the brain. So now we replace that ligand with a drug that goes there, keeps the protein stable, it doesn't dissociate, and it doesn't aggregate. So that's what we call native state stabilization. The challenge with synuclein is everybody thinks that synuclein doesn't have a structure, and therefore it doesn't have that lock and key fit principle.
So you can't keep it in a structure which keeps it happy.
But in reality, it's not true, because synuclein is structured. Synuclein, once it's bound to membrane, is structured. The issue is we don't understand enough about its structure in membranes and what regulates its dissociation in membranes. And that's the work we're doing now in trying to figure out the best way to keep it in its normal state.
Okay, so try and keep it happy, try and keep it in the state that it was always intended to be.
Exactly.
And therefore, if you, as I could see that, so if you keep it there, then whatever happens after that, it doesn't really matter where it goes, because I've stopped it going down that path. Okay. Well, I mean, this is an extraordinary story, your story. I mean, first of all, your personal story is extraordinary. We haven't explored it in great detail, but I mean, just the notion that you arrive in New York and you have to create a life for yourself is extraordinary. Not only do you do that, but you do it in such an extremely generous and fabulous way with your science, really. I mean, it's been an extraordinary story of how you've taken chemistry into biology and biology back to chemistry and into the clinic. So it's an extraordinary, exciting voyage that you've taken us through. I would like to thank you very much for making it accessible. I find a lot of people, when I talk about alpha-synuclein and structurals and aggregation, I get a bit lost in all of it, but you've been extremely clear in describing it. And I love the idea that you have, a vision of where this will go and how we can actually make a real difference in this condition, and not just view the fact that we have to use antibodies to treat it. If they haven't worked, it doesn't mean it's not relevant. So thank you very, very much.
Thank you very much. Just want to finish by saying that it's a story that's been made with the help of so many people along the way, and I'm grateful for all members of my lab, present and past, who really did all the work and deserve all the credit. Thank you for having me. And
well, thank you very much. There's just a few other thank yous whilst we're here. Just before we sign off, I'd like to thank Supernus Pharmaceuticals for supporting this series. Without them, we wouldn't be able to do it. Our next Research Spotlight takes a very different line. So on October the 21st, we'll be chatting to Lynn Rochester from the UK about walking and health in Parkinson's, which will impact on the native and happy state of alpha-synuclein. So listen to that. And I want to remind everybody that if you want to know more about what goes on in the world of Parkinson's, you could go to no better place than Phoenix, Arizona next May. We'll have the next WPC. So with that, I would like to thank you all very much for joining. I'm sorry we didn't get to everyone's questions, but you've been a fantastic audience with some of the questions you've thrown up. Again, I would just like to thank Hilal for a superb discussion on something which lies at the heart of Parkinson's, giving it a wonderful, wonderful account of what might be going wrong and how we can deal with it in the future. So thank you all very much.
Thank you very much.
I wish you all a good day, good night, or wherever you are in the world. Bye-bye now.